Ever since Darwin, biologists have understood the importance of the tree of life metaphor. In Philomath, we will learn how to infer that tree and how to use it to understand biological processes. Philomath is made possible through a career grant from NSF, as well as ongoing support from the University of Tennessee, Knoxville.
All right, so let's start class. Um, so this week was about building trees, and I had you just do RaxML, right? We're going to talk about Bayesian stuff later on, and so I don't want to dig into like Mr. Bayes. I think we're going to talk about dating, where you build a chronogram, and parsimony. You know, it you can think of it in terms of like as tree inference, it's not harder than likelihood. I don't think I did the model selection and stuff like that. Parsimony is simpler. Um, so I'll talk about any issues people had with doing the tree search stuff, and then we talk about the papers too, and other issues that come up with tree search. Okay, so have an active discussion about it. So first of all, the RaxML stuff. So what I wanted you to learn from that was first of all how to go through a manual and figure out how to run stuff, right? Because RaxML has a ton of options, right? And so to figure out what to do, you have to dig through and learn how to skim the manual and things like that, right? And think about what the models mean and stuff like that. Right? So are there questions about that? I know there was still some issues with like installing it on Windows, right? You had that issue. Um, do you?
I actually was able to get it really easily because I knew how to do the system map.
Okay.
I figured out how to do it back.
Mm-hmm.
So that was all I had to do was add it where that folder was.
Yeah.
There's probably some more elegant way to do it, but I just
Buying a Mac.
aside from that,
So what were your issues with it?
I just couldn't figure out. I was trying to move the path and it just moved the error messages.
Okay.
So
Did you get to work? Okay. Yeah.
I'm having issues too. I don't even know what's going on.
They're farther down the line.
Yeah, was downloading the package. I was getting some really weird error, and I tried to refork and do it again, but I'd already committed, so I just downloaded what I already had. And so
Yeah.
I tried to download your file, but I'm getting some really weird errors.
Okay.
in the terminal.
Okay. So we just like break off later and have a Windows support group, and yeah. Okay. Well, I mean, did the Mac people have or the Linux people have issues? Did you get to all download and install?
I mean, everything installed, but he came up with some little
Oh, I mean,
argument for.
so farther down when you had us install the package that we fixed, right, and then plot the tree in R.
Yeah.
So when you direct R towards your data files, right, you have the default for in place is false, and you can point it towards a directory.
Mhm.
And I was having some like permissions problems with that, so I just set my working directory to that directory
Yeah.
and set in place equals true.
Yeah.
So it's a workaround. I don't know if that's the right way to do it, but
Yeah, and so the issue, I was saying, so normally you have in an R package you have files that stored and stuff. You have like regular R data files. Those are in the data folder in a package, right? So here, let's look at a sample package. Let's go ape. Okay, so here's ape. And so we have data folder, the R folder, the test folder, right? And so normally data goes in here if it's R data, like RDA.
those are like sample data that come with the package.
Yep. Now those typically used just for stuff saved as like R objects, right? If you have raw data, typically it's not stored there, though they actually do. Looks like typically that's stored in inst and then X data there, right? And what happens when you actually install a package, it moves that internal folder X data up a directory, which is odd. So that's one thing it does. However, to test that package we use for doing RaxML, for testing RaxML, actually does it before you start moving stuff around. And so it's trouble. So we'll test that. What I did was just put the directories, the files in the same test.dat folder. Right? And this took me a couple days actually to debug because it's just annoying and a pain. In real life, of course, you'll have your test data somewhere outside your package, right? So this won't come up on your desktop or something like that.
So I just set the working directory to inst x data.
Yeah.
And then set in place equals true.
That's good. Yeah, that's a good workaround. Yeah. Because when I did the solutions on my computer, they worked. But the permissions thing, I mean, depends on where you downloaded and installed it. And Christian, you have permissions things sometimes because you're like used to be a different user, right? So,
I got it to work though.
yeah. So permissions in general, what are they for on a computer? What does that mean? Because this is something that will come up periodically, or you'll be told to like chmod something, right? And I'm going to go dig deep into it, but does anyone know what that sort of means?
If there are multiple users, does that restrict access to some of the users?
Exactly. So on my computer, we only have one users here. Yeah. So my computer has 3 user accounts: guest, shared, and Bomera. You also have, your spouse's account or something like that. Or if you're on a cluster, there's tons of users. And you want to make it as such that, I can edit my files, but I can't edit your files, right? There's some files maybe we all want to be able to read, right? So maybe I want to be able to read, how to use this server and put that somewhere. But I don't want to let Orlando read my letter of rec for him, right? Like, he has too many swear words in it or something, right? And so you can set different permissions of like, anyone can read this, or we have a program I want people to use, anyone can run this, but no one can open inside it, anyone can edit it. And so you can set those permissions up. And your computer actually has permissions too. So we maybe doing the RaxML install, we had the sudo thing, right? And why do we have sudos? Because the computer doesn't want you accidentally messing with core functions. Like if you took the ls function and accidentally stored your shopping list there instead, right? You didn't really ls anything, you just have a shopping list every time you try to look at stuff, right? And so you have to know what you're doing before doing that. So that's why it has all these permissions. So in your case, you probably had, the folder was owned by someone else or something like that.
I tried setting,
Yeah.
Outside of R, changing all those permissions so it could be accessed, and I still couldn't fix that.
Yeah.
So, I don't know what the problem was.
So there's 2 ways so you know what to do. Just for people in general, what you can do is, let me go to my documents. This thing you can do, you can take this and get info on a Mac, Command I, and then you can change permissions, right? Or if I do ls minus, I forget which one of these shows permission, but which one of those flags to ls has it show me permissions, but it's one of them. And this tells me this is a directory. I have read, write, execute access. Other people on the computer have read access and execute access, but not writing to it. All right, other people here can read it, but not execute stuff. Right, and so sometimes you'll see that like rather than you owning it, it might be admin owns it or something like that, and you can't get in there. And so that's one thing that's one little hiccup that can come when you're trying to install stuff. Okay. What else people have issues with?
Like the input and output paths, like figuring out where things were and where you would put stuff.
Yep.
Yeah, I ran the functions without installing the package after I fixed the package.
Uh-huh.
And so I had all these
Oh, yeah.
documents that were saved in the folder where I told it to, because I had to explicitly say the input and output.
Mhm.
And then that's why I had trouble trying to do further down the line, install the package and stuff. So don't mess with the package before you install it.
Or after you mess with it, install it again.
Yeah, so
Yeah.
that's what I did. I just recloned and started over.
Yeah. Okay. Yeah, and one thing about test that is when it creates new files and stuff, it doesn't clean up after itself that well. And one thing I actually did when I was writing this package was make it so it would store output files by default on your desktop. Right. So when you run RaxML, it's good to see what's actually happening. Right. So that way you can test. You can look at what the files are. Right. So questions about that? Seem good. Yeah.
As a general question, so what's the advantage of, for example, using RaxML for R like this?
Oh, often none.
Read a function and
Yeah, no, I mean, it's basically so for the class, you get the whole same framework of I'll modify stuff and can check it easily. In the real world, you often won't do that, or actually, I mean, what I've done sometimes is have a little R script that will like create one folder per gene or something, and then run raxml for each gene, and then store that. So you do that, then you are doing system calls, but you're not writing a function to it. You're just doing system raxml -fa. Blah, 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 blah. Yeah. And doing it that way rather than sort of on the fly figuring out what do I type each time is better because that way it's reproducible. Now a trend in biology for some people start making make files, which are these complex things that tell you, you saw it when you're making raxml, right? You downloaded it and you type make, right? Instead of doing all this stuff, right? So like .o. What it was doing was compiling it. And what makes does it's nice is it can already see if the output for one step is already there, and if so, it can skip that step. So if you were doing a whole bunch of runs and everything works except for your last gene, if using make, it will go through and say, okay, that worked. Oh, then do this gene, I'll run that gene. And so it makes it reproducible, and then you don't have to restart it each time. Right? That's still techy enough that you shouldn't bother with that. But having a basic script of what you do each time is really handy. Right? I had a paper where we ran 19,000, I think, BISI models, MUSI models on data, and we found out that one of our characters was a bad character, that it wasn't biologically meaningful, and so I would have had to redo all the models. All I do is change, s equals 7 to s equals 6 characters, drop that last character, and resubmit, and it all ran again. Another, 2 degrees of global warming temperatures, whatever. But it was one change, but it's reproducible. And also, people who want to go and check your work later or build on it makes it much easier. Right? So that's why doing some sort of scripting is nice. And I mean, in this class, we're doing a lot of R. But you can also do Bash scripts or Python or whatever. Other questions?
Are we really more closely related to frogs than we are to cows?
No, not most of us. There's something called when it comes to integration, probably.
So I think my question is similar to some other people had. So I already had raxml installed on my computer, so I don't know. So it's installed in my users folder or whatever. So
in like user local bin or like users your name?
Users and then my name.
So unless you add that to your path, it won't see that. Right? So I mean, like you have like your find file utility on a computer, right? You can find that will search all the directories for a file, right? And that can take a long time. When you actually finding a program to run, the computer looks in only certain places, right? So my computer, that's where it looks. So it's directories separated by colons. So opt local bin, that's where MacPorts put stuff. User bin, that's where your computer's like basic programs are stored and things like that. And so what the computer does is if you say ls, it says, okay, this guy probably wants me to do some program. What program is it? So it looks here. Is it there? Nope. Is it here? Nope. Is it here? Nope. Is it here? Yep. Okay. Now it can be a very different version of ls than what's here, right? Because this one's earlier in the path, that we'll use that one. Okay. And so it doesn't have to search your entire computer. It searches in certain places, right? And so in your case, unless you tell it to add, users, whatever your short username is, applications, whatever, it won't look there. Yeah. Which is why I had you do sudo cp user local bin. Yep. Does that make sense?
It does those in order, in that order?
Yep. And so, like for example, like MacPorts installs newer versions of things, right? Versus what Apple installed. And so the MacPorts version of Top might be better than Apple's version of Top. That's probably a year younger and has better flags or whatever. And so it will run that one instead. Yeah. Okay. If you don't want to do that, you can just pass absolute paths. So I could do /users /biomera /phylogenic program /raxml /raxml HPC -fa whatever. Right, but it's a pain in the neck to do each time. And so it's better to just put it in your path. Okay. And if you use Cygwin on Windows, you also have paths. I'm not sure how regular Windows command line uses paths or not. Anyone know?
I wasn't able to do any of that in command line, but the paths, where you go into the system options, and you can go to some advanced options, and under your system properties, you can do environmental variables, and then you can mess with the path and in your list, and you can just take that folder you want to add in, put it at the bottom of the list, and it'll find it. I had trouble with, I tried to put it up at a higher level and see if it would search the folder below, and it seems not to be doing that for me. So for this program and for math that we used, I had to actually put the full down to the folder where it is, where the first binary is, or
Right.
the first application.
Yeah.
But that's an easy enough workaround that I don't have to worry about it.
Yeah.
Like I said, it wasn't much elegant.
And on a Mac, that's one thing where which command we used when we were doing math last week. Remember we had you do which, and one reason for that is if I do which raxml hpc, it tells me which one it's using. It's using one in user local bin raxml hpc. Now, as the Mac ports version of raxml, that's actually a version old, so it's like raxml version 7 rather than 8. So if I installed that, then it would use the ones in Mac ports, which is the older version. And so I don't know what's going on. Why is because the commands I had you use, some of them are only in the newest Mac XML, right? And so I would have had those fail. I didn't know why. I could then look and say, oh, wait, it's not using the one I just installed. It's using an older one somewhere else. Does that make sense? Now, there are other places you can run it beside your own computer. I mean, so we have a cluster on campus. There's various other clusters. I've used this in the past, Cypress. So this is from an old NSF grant, and it's being supported by, I think, San Diego Supercomputer Center right now. And this is a science gateway. And what it has is friends like Raxml, Maft, Paup, Poi, Garley, Mr. Bays installed for you. And you upload files there and tell what you want to do. And pull down, I want you to do a run with bootstrapping, 1,000 bootstrap reps, so forth. And it'll send it off to supercomputers and run it for you and then give you back the results. Right? So that can be better than doing it on your own computer. And they've actually tweaked the code so it has things like it can run in parallel. It can do checkpointing, and you can stop halfway through and then pick up again. But it's also using an older version of Raxml. I mean, the ones you installed was 8.03 or something like that. This is 7.27. So some of the stuff we had in there for like morphology, I don't think is on this version. Okay. Yeah.
So if you run something on Raxml, so I have like 15 genes put together, and then they're all partitioned, and I have a guide tree. It works on my computer, Raxml, but it doesn't work on Cypress. It just returns nothing. So is that probably because it's using an older version?
Yeah. Yep. And you could email them, and they're very friendly. And you could say, hey, here's my issue. I mean, one issue is, they have people who are on staff, but they don't really have a lot of money for running this now. So, if someone's being paid 2 hours a week to work on this stuff, it might take a while. But they really do want people to use it. And we actually have an R package interfaces with them. I think it's on CRAN. It's running RForge. So you can run this directly from R. Try that. Yeah. So let's talk about the partitioning a little bit. So how did you do that in Raxml, right? Why is that a useful thing? What does partitioning mean in this case?
So it's just you separate genes. So like if you have 100 species, you'll have all of the data for that gene for all of those species partitioned into one thing, right?
Mm-hmm. Right. So you can do by gene. You can do other ways too. So I have, here's my super matrix. right? So they have, missing data, this one, missing data, that one, and so forth, right? And what you can do is say, you use one model for this and one model for this, right? Why do you want to do that? And more models, more complexity, more time.
Maybe for a particular taxa, you only have a handful of genes.
Mhm.
And like if you included all of it, it wouldn't be as useful as if you only had a subgene.
Not quite, because we're partitioning here by gene, not by taxon. So you can imagine partitioning by taxon. So like, woody plants have different rate matrix than herbaceous plants, right? And that's not done yet for tree inference. With one of the things, it's called P4 can allow you to do that, but no one uses it. That's done very rarely right now. So it's not by taxon, it's by gene or by site. You do like first, second, thirds, or different partitions. Right? Why do we have models? What's the point of a model?
The different genes might have different rates under which they evolve. We can take them into account if we separate them.
Exactly. So we have models to help model reality, right? So with likelihood of trying to fit the data well, right? So if I have different sites that are evolving very differently, if I have one gene is very sensitive to GC content, right? Has a very low GC content, and another gene doesn't, right? Well, if I have two different rate matrices, one might favor high GC content, one might favor low GC content, right? Question? Okay. And so I can have them all be the same rate matrix and have something intermediate, or I can have two different ones that will fit better, right? I can have a slow gene and a fast gene, right? So I can have CO1 and 28S, right? And I could say, rather than having them all have the same rate of evolution, I could say, okay, CO1, you have a fast, you can have whatever rate you want. I'll choose fast. 28S, you can have any rate you want. I'll choose slow, right? And let them fit that way. Yeah, question?
So the other thing, I haven't used this, but the other thing too is that if you're using like protein data or RNA stuff, you can use a different model for the first two bases in the codon versus the third base in the codon.
Yep.
Because you expect the third one to evolve.
Exactly, right? Because of the silent third mutations, right? So you can segregate it that way too. Yeah. Or you can also now do partitioning where you have, DNA data for some sites, amino acid data for other sites, morphology for other sites, right? And so then other characters, and so then you can fit the right model for each one, rather than assuming that morphology is like DNA because of how missing two characters, two states, and things like that, right? And so why do you want to use morphology?
It might be the only data you have for some taxon.
Right. Why?
I don't know. They're a museum specimen that you can't get DNA from or something.
Mm-hmm. Museum specimen, fossil data, right? It's very hard to get. There actually is T-Rex DNA, protein in GenBank, but it's like a short little string. But yeah, it's pretty hard to get, theropod DNA of birds, stuff like that. So for those things, you need morphology. If you're trying to date stuff, right, put a fossil on a tree, how do you put a fossil on a tree? We'll get to this when we get to the dating week. You just need to say, this fossil, here's my tree. This fossil attaches, somewhere on this branch, say. How do you know that, right? And sort of like, traditionally we just sort of eyeball and say, oh yeah, it's an aster, so it must be, somewhere between the asters and the sister asters, it must appear there, right? Well, how do you know it's, that's just using the taxonomy. And nowadays we build trees using data, not just taxonomy. And like, yeah, it seems like it's that clade, right? So you can then, do an actual phylogenetic analysis where you have your molecular tree, and then you have the fossil goes somewhere, right? And this isn't a new idea, but people are acting like it's new in the field right now, where putting fossils on trees using morphology. So it's rather than just eyeballing it and sticking it in places. That's something you'll see a lot. People are using Beast for and things like that. Actually, PhiTools now has a function called Locate Yeti, where If you have some taxon you only have morphology for, a continuous trait, it'll just use a method for a continuous trait evolution to figure out where that goes in the tree. So if you have some morphology from Bigfoot or something. These are for putting an extinct anolis on the tree. And again, it's not really new at all. It's just people are now doing it again. Right? We've made trees with morphology for years. And so by partitioning, though, you can have all the data talk to each other. Okay. Other thoughts about partitioning? Oh, you could do that. So like if you use Palp, right, you can do, I forget, it's been so long, is it delete or drop? What's the command? Like one's removing tags, one's removing sites. But yeah, you can, if you have this partitioning knowing where boundaries are, you can say, that's what happens with the tree of just mitochondria, right? So like Sam in my lab's working on fish evolution, right? And fish apparently have sex like mad with other species, and so mitochondria is slipping all across species boundaries, right? So you might be curious about, does the mitochondrial tree match a nuclear tree? So I have a matrix that has both of them. What I can just do is throw out the nuclear data, run it with just mitochondrial, throw out the mitochondrial, run it with just nuclear, and compare the topologies. Right, so that's useful for that too. Yeah. Good. And we'll talk next week about ways where you can use that different tree information. So with what we've been doing this week, we take our one matrix and we assume that there's one tree for all those characters, right? So we're trying for the species tree, right? Now in reality, of course, different genes have different histories. We're talking like mitochondrial introgression. Right? The mitochondrion then has a different history than the nuclear genes, right? With RAXML, it assumes it's all the same history. It's trained off of that same species tree, right? I'll talk ways about relaxing that next week. Good. Other questions or thoughts about partitioning? Okay. What about we can do in RAXML, which is just seen by reading the manual, which I didn't have you do though, using a constraint tree. All right. So what is a constraint tree?
It's a quote unquote known tree. So that'll help you get to the quote unquote right answer. So like for the tree I'm making right now, it's like it's a bunch of things scattered everywhere, and I really just want branch lengths, but I also want like topologies within families worked out. Whereas I don't want like gymnosperms like nested somewhere in angiosperms and stuff. Things are fairly well resolved.
Mm-hmm.
Since my data is smaller than what I use to resolve those things, it might make a weird answer or something. So it's kind of weird.
Yeah. So it's cheating, so that you always get the right answer if you're afraid of getting the wrong answer. If you don't trust your data, you still want the right answer, you can do it. Right, so you have cases where, so we know, like, monocots are a clade, mammals are a clade. We got that, right? And you think about the size of tree space, if I'm doing a tree of vertebrates, right, looking overall at whole tree space, it has some things that have mammals not clade, some things that have, reptiles and mammals, we have birds, And so why look over that space when I know much of the constraints of that? So what I can do is have a constraint tree and say, okay, mammals must be a clade in this tree. So only look at those trees that have that. Now within the mammals, it could flip things all around, right? But it keeps it all constrained. And so people use them. They can be a little controversial because, we're oftentimes wrong, right? And so you could say, have this constraint tree, but then with more data, put the wrong answer. But, we find, if I take that constraint away, keep getting this consistently this other thing. That might be right. Right? So ideally, you don't have to use a constraint tree. Right? Your data will be enough to tell you. And it's also a good way of seeing how good your data are. Right? So if we know monocots are clade, my data is saying monocots aren't a clade. You say, that seems weird. And it could be my data are for the wrong scale question. Right? Maybe I have really fast gene, and so the time I get back to those relationships, it's all saturated. All the information has been lost. But also you have some introgression, you have something labeled wrong. Right? You might miss that if you keep the stuff you think constrained, and then you miss that, right? Because there's stuff in GenMake all the time that people, sequence the gut flora rather than the tissue that it was meant to, or just have contamination in the lab or something. And so if you constrain it too much, you wouldn't pick that up. You just find some really weird long branches and really exciting relationships.
But so you could use a constraint tree to test, like, see how much your data supports specific hypotheses.
That's true. So if we're thinking of decay indices, right? So this is a parsimony thing. We don't use much within molecular stuff anymore. But what you can do is take your best tree. I'll talk later about a little later in today about ways to measure support for this, right? But when you can do support is say, I have 3 characters changing on this branch. Fine. But it could be if I have A, B, C, D, E, F. It could be if I have, A with C, B with D. I now have, those characters separated somewhere else, but now I have other characters changing, right? So it could be that, yes, it has a branch length of 2, but only one of the characters is actually supporting this. Right? So what decay index does is say, all right, we have this best tree. Now find me all trees, the best tree that does not have this relationship. Right? So I have this, CD versus A, B, E, F. Right? Find me trees that don't have that. So it can have, it's called a reverse constraint. Right? How much worse are those trees? Is it 1 step worse? Is it 1,000 steps worse? I actually, for part of my dissertation, I worked on a likelihood equivalent of this. We see, like, for the different genes, is it supported by its first codon position, the second under likelihood, right? So you can do that sort of thing too. So there's no use of constraint trees. Yep. So they're not used much anymore. I mean, sometimes for like large-scale analyses, there'll be a constraint tree, right? So we had a paper published that had, it was like 50,000 species of plants. Maybe it's 45,000. A lot, right? And that was on a constrained tree search. And even so, some relationships on that tree are really pretty bad. Like Amarillo is nested up within something else. It definitely has issues, right? But that's because it's a very sparse matrix. I mean, some genes are common across many plants, but oftentimes genes aren't sampled across everything. And so to help make sure we got the right answer, because our question wasn't really with the tree. The question was mapping traits on a tree. We had to get a tree anyway. So we made this tree that sort of constrained to be what we know. Still wasn't great, but better than it would have been had we just had no constraint tree.
So is rooting a tree not just a kind of a constraint tree?
So rooting, so that's interesting. So for most models, rooting will affect the score, right? So people know what rooting is? There's a video on this, but sure. Who's a little fuzzy on rooting? Okay, yeah, so rooting. So I mean, with parsimony and with likelihood and everything, if you have a time reversible model, right? Right? If I have, this is state 1, state 1, state 1, state 0, state 0, state 0, right? It's going to be a change in this branch, right? Under parsimony. Under likelihood probably too, but not necessarily. And that's the case no matter where I root it. If I attach the root, here, or here, or here, the total tree score won't change. And so in your phylogenetic analysis, you actually don't have any information about rooting. An exception to that is if you have a model that has like a clock model, right? Where if I have a tree that's, has these branch lengths, right? If I instead rooted it here, I would have, this really weird short branch, and then a longer branch, and then, something like that, right? Where there's no longer an ultrametric tree. So there, the rooting could matter in terms of the tree score. If they have to stretch this branch more and such. But in most models, we don't have a penalty for that sort of thing. So what you could do, though, sometimes people make the out group, make the in group a clade or something like that as a constraint tree. Right? There's nothing. So the way we root trees often is by attaching the out group, right? Something that's far enough out that we say, we know whatever the relationships are in this group, you're not part of it. You should be out over there. That's how we do the out groups. Now, when we do that, we could do a constraint tree such that we enforce that belief that the in-group's monophyletic, right? Or we could allow it to vary and just see if we get some weird errors, right? the trick with outgroups is they have to be similar enough that they're still relevant, right? And have the same genes and stuff, not saturated, but not so similar that you might actually be wrong. That might be in the in-group. That's why I'm doing a tree of plants. I'm not going to include humans as an outgroup. They're just too far distant. I could include green algae. So more questions about that. Actually, the Open Tree of Life project we talked about last week, when you load your trees into there, they have you specify what the in-group is, and they only use the in-group for figuring out relationships, because the trees often have multiple outgroup taxa, so you have some sort of relationship of those outgroup taxa. They're thinking that you actually don't care about those taxa, so it might be very bad relationships, you have bad genes for inferring them and stuff, so you toss those out in the synthesis. It's a bit of a pain because you have to actually navigate through and like on a multi-thousand taxon tree find the in-group node, but it's good for the synthesis later. Other thoughts or questions about this? Okay, we talked about partitioning, we talked about rooting, we talked about something else. Okay, indices and things. What about tree search itself? Are there any questions people have about how the tree search actually runs? Right, so I know people use PALP in the past, right? And so there's TBR and SPR and NNI and all those acronyms, and RAXML has other ways of optimizing stuff. Do people know what all that stuff means? Yeah. So I gave an undergraduate exit seminar to guide the thesis. And all my friends are giving seminars. They're all grad students. I give a seminar, and then I said, I built this tree using TBR, and someone said, "What's TBR?" I said, "I don't know." She said, "Avoid TLAs. They're acronyms. You don't know what they mean." So it's worth knowing what this stuff is. Basically, these are ways of moving across tree space, right? So think about we talked about how big tree space is, right? And I could just pick trees at random from that tree space and have that for my search algorithm, right? That's dumb. Why? Space is huge, and then if I'm in a good place, the next one I choose could be bad, right? It's just a random draw, right? So you want to sort of choose things that are sort of similar, right? So remember the video I talked about like boxes of chocolates. If I like the chocolates that have green sprinkles on them, I'll try the ones that have other colored sprinkles rather than something completely random, right? So I want to get trees that are sort of similar but slightly different. And so what we do is take a tree and break it and modify it in certain ways. Rather than trying a whole new tree, I'll take off one clade and try sticking it somewhere else. Right? That's called a subtree pruning and regrafting. Right? Or I'll take, a branch on the tree, this one, and swap this position with this position. It's called the nearest neighbor interchange. Right? Or I'll cut the tree in half and then draw a new line from any branch on one subtree, to any branch on the other subtree and make a new edge that way. It's called the tree bisection reconstruction. Right, so those sort of moves you go through, and they allow you to move across this tree space and sample it. That make sense? Okay. And there's other kinds of sort of cost savings. So RAXML is much faster than PALP, for example, for likelihood. Right? Why is that? Because they're both written in C. Right? They're both written by really good coders. Well, RAXML is sort of sloppier than PALP is for branch lengths. So PALP, if you propose a new tree, then it optimizes all the branch lengths. Right? RAXML optimizes branch lengths in the local area that you've modified. It's like this new pink branch and stuff attached to it, but the other stuff doesn't get modified as much. Right? So it saves time that way. And it can later do sort of an optimization step if you want it to. What can be a downside of that, though?
There could be a modification where it has a better likelihood, but it necessitates like changing branch lengths a lot.
Right.
You miss that.
Yep, so you'd miss that. Exactly. And so all these algorithms we have, for once you get over 16 taxa or something, are called heuristics. Right. So remember the video we had talking about like NP-complete problems in the XKCD cartoon, right? So There's no known algorithm that is guaranteed to give you the right answer for finding the best tree under parsimony or likelihood or things like that. And so we have all these ways to get to hope. We hope we get the best answer, but it's not guaranteed to, right? And so depending on which approach you use, you could miss many good answers. Okay. There's also things called islands. So if you're thinking about swapping trees, it could be that I have this tree, all the ways to break this in one step have worse likelihood. Right? But it could be that some trees that if you did 3 rearrangements, they're way better. Right? But if you have an approach that's only greedy, that only goes uphill, it can never get there. Right? And so there's different ways like simulated annealing and things like that allow you to move across tree space in different ways to hopefully escape those islands or start from different trees. So there's many approaches. This is actually a big issue with tree inference is that you can often get the wrong tree. And given the size of tree space, you almost always must be getting the wrong tree. Right? When you look at a small fraction of trees, and I mean we tend to get trees that agree with what we know. Right? It's not completely random, right? But it's a very, very, very hard problem. So it's worth being cautious. When RaxML runs fast, like, hey, it's done, right? But it could be giving you wrong trees, right? And so it's worth being cautious about that.
But so for like we had, 9 sequences for the DNA one?
Yep.
I mean, the space is a lot smaller, so how accurate or confident can you feel that the tree made by RaxML is?
That's probably going to be a fine tree. I mean, but it's still, what you could do is you could do an exhaustive algorithm, right? So you could try each of the possible trees for something that small. Take a while, but you'd be guaranteed to get the best one. You could try a branch and bound algorithm that is faster than an exhaustive one and is guaranteed to get the right answer, right? But those don't scale well. So if you look at like how long things take to run, it scales faster than linearly with the number of taxa. So you have to start using approximate heuristic methods. Other questions about tree inference? This used to be a really hot topic. People come up with new, I remember when the parsimony ratchet came out. It was a way of doing parsimony quickly, and it was way faster than what was out there before. People were really excited about it and stuff like that. Now, RaxML keeps improving, but it doesn't seem to be the same excitement about this. More of the excitement seems to be going into the ways that deal with, gene tree, species tree conflict. We'll talk about next week and stuff that way, right? Or next new kinds of sequencing data. Stuff like that. Okay. Bootstrapping. So one of the readings was on bootstrapping. And you've probably heard of bootstrapping before. Right, so why do we care about bootstrapping? How many of you have bootstrapped for a tree before? Okay, about half of you. Why did you bother doing that? And what makes you think you can trust it? What does it tell you? How does that work?
If I can trust it?
Yeah. And what
if it's above a certain percentage, you know that everything in that group has a higher likelihood of being together as opposed to
Right. And what's sort of the underlying idea behind the bootstrapping? Right, so basically it's a robustness test, right?
Yeah, philosophically, it's also assuming that there are things you couldn't have sampled. And so it's actually creating instances where things aren't sampled from your data and seeing if the other data is affecting that or not affecting that.
Right. Yep, exactly. So it's sampling with replacement from your original data. Right. The idea here is you're approximating if I went out and got a whole new data set, what would it look like? And we don't have a way to get that whole new data set without going out and doing it. So we're pulling ourselves up by our own bootstraps and using existing data to make up new data sets.
Is that really why
Yeah. Yeah. I mean, yeah. People use like same way we like Monte Carlo methods because people go to gamble at Monte Carlo, right? That's why they name things this way. So, yeah, so it's picking yourself up by your own bootstraps. That's why it's called that way. In phylogenetics, we typically use non-parametric bootstrapping. Right? And so the Felsenstein paper you read was about non-parametric bootstrapping. So what does that mean? What's a non-parametric method? I'm going to call Jim. He's going to come get you.
It uses rankings instead of parameters.
It doesn't say ranking, but yeah, it doesn't. It's not a model-based. It's not a parameter-based model, right? So basically, what it just does is sample with replacement. So you have a list of 10 sites, and I just draw 10 random numbers from that. I can draw 1, 3, 1, 4, 6, 1, until you get another 10, and then you just rerun your analyses the same way. Okay. Now RaxML does something faster and more approximate than that bootstrapping. It stores like different sites already, which is why it can actually do bootstrapping for giant trees without taking 100 times longer than doing the first tree search. Still approximate. And actually, the paper showing that the actual bootstrap we all use is also an approximation that's also biased. Right? The opposite of kind of bootstrapping is called non-parametric. It's called parametric bootstrapping. What do you think that would be?
Also uses parameters.
Yep. And so, can you give any examples?
Like you resample from like a distribution described by parameters.
Yep.
Rather than from the original data.
Mm-hmm. So a great example of this is looking at long branch attraction, right, which is also one of the readings this week, right? And so to look at that, what you can do is say, okay, if my tree were actually this, would I come back and get my original answer? So what you can do is make up your new tree, evolve data on that tree using parameters that's made from the original thing, and then infer the tree from those data. And then you can see, do I get my original tree back, or do I get my made-up tree back? Right? And if long branch attraction is an issue, you could get the original tree back even though you made it up under a different tree. Right? So that's use of parametric bootstrapping. Helsenbeck had a good paper on that. Okay. So what tree inference method did we use bootstrapping for? So if I'm making a tree with parsimony, could I use bootstrapping? Yes. Yeah, all the time, right? Because it's a good way to estimate certainty. But also do other things like the K index I talked about, right? That's typically a way to do it. What? Yeah, so jackknifing is like bootstrapping, but instead of sampling with replacement, you sample without replacement. You say, okay, let me take randomly 70% of my data, see what I get. Let me try sampling 30% of my data, see what I get, right? And I'm not that keyed into this literature. I mean, I believe there were tests showing that bootstrapping is a better estimator. That said, there's a group of people in phylogenetics who do jackknifing rather than bootstrapping. I'm not sure why they do.
I've just seen in paleo they'll do both and get numbers.
Yeah.
Which is better.
Yeah.
So if you have 10 characters that you're inferring the tree from,
mm-hmm.
and you use bootstrapping, redraw 10 characters with replacement.
Yep.
So the same number as your original.
Right.
So how does it compare? Like, how does your interpretation of bootstrap values change if you have 10 characters versus 10,000?
Yeah, so the probability of a given character being sampled actually asymptotes pretty quickly. And so basically, if you're thinking of just plain parsimony world, right, in order to have an edge supported with a certain amount of support, you think like, what's the chance of choosing 0? So say there's one synapomorphy for a clade, right? What's the chance of sampling that by chance given its bootstrapping? And it's, I think like 1/3 or something once you get large. And so you could say, okay, like in order to recover 70%, you need to have 3 characters or something so I can sample them all. So that you get that asymptote. Now at small numbers, I'm not sure how it works. I mean, you could easily do a simulation and say, trait A, how often do I sample trait A? And basically, it's a flat multinomial. Yep. So we do bootstrapping for parsimony. Do we do it for likelihood? Yes. Yeah. Yeah. So if you see a tree published with likelihood, we can see numbers below the branches, below the edges. Those are bootstrap values. Okay. What about Bayesian analysis? Right. We do posteriors instead. Can we do bootstrapping?
Because that's just the way you're sampling your data.
Right. So you could. Right. So we could do bootstrapping with Bayesian. Right. And so I have a video posted a couple days ago, I think, about Bayes versus likelihood. Right. And with Bayes, we only report the posterior, but you could implement a bootstrapping procedure with Bayesian stuff. So you could say, for my Bayesian analysis, I take the tree of maximum posterior probability, write that topology, and then I can make up a new data set, run it again, so you get the same topology back, and keep doing it, right? And so I could bootstrap Bayes. And so I would have, my bootstrap estimates of the clades and the bootstrap, and you could also get confidence intervals and, well, not confidence intervals, but bootstrap proportions for posteriors too, right? No one does that. Partly because it already takes a long time to do a Bayesian MCMC run, right? So if you add bootstrapping on top of that, it makes it 100-fold slower. So we want to be efficient. They also both feel like they're giving you the same information about support, but actually they're not, right? With bootstrapping, what matters a lot is how many times I have a trait there. So I once did an analysis where I had like this. And so forth, right? So I had one parsimony informative character, right? So this says A, B, so the tree it would form is A, B, C, D, right? That one character. Nothing else changes, right? Now with bootstrapping, as I increase the number of these invariant sites, what will happen?
Less likely to take this down.
Right, so that's something. It's asymptotes, but pretty soon, I have a certain almost constant probability of sampling this, right? So I'm drawing 1,000 out of 1,000, so it's just sampling this one. It becomes pretty stable, right? So the bootstrap value does not get super high. Right? It's pretty low for this, but it'd be like 33%, right? No matter how many invariant sites I have. What about Bayesian approaches? What would happen there? What would happen to your posteriors? Well, would your analysis ever not have this character? No. So that's the difference right there, right? So with bootstrapping, sometimes you don't see that trait, right? In some of those replicates. Bayesian, you set the data set as they are, as it is, with no changes at all. So if there's just one character there, it always sees it. Right? So what actually happened here is you go from very low support, if I have just one trait, as they add more invariant sites, the posterior probability shoots way up. And what's happening is it's saying, oh, these data evolve very, very slowly. Right? They have all these invariant things. So the fact that I saw something happen once, saw some change, it's far more likely that happened once on this tree rather than twice on this tree. And to see how slow these data are, right? And so your posterior shoots way, way up to 100, right? But then your bootstrap stays constant, right? Because they're measuring different things. Bootstrap is saying you're looking at your robustness, whereas posterior is looking at, given the data you have, what's the probability of seeing that result, right? So you do different things. Okay? So you could do both, but no one does. So anyway, if you want to create a new method at the end of the class, one thing you could do is, boot Bayes, or something like that, and run that.
So is it a problem to have non-informative data if you're making a tree using histories, for example?
If you have only non-informative data, yeah.
Okay.
If you have some, though, that's fine. Because if this is like, let's say you have nucleotide data, right, and some are invariant, some aren't. If that's actually how the data evolved, then that's fine. It's important data. It's important information for the model that the rate of evolution is not super high. If all I had was the invariant site, the variable sites, then it tells the model we have having lots of changes here, so there must be a high rate. So that any change I see it could have happened once or twice or 15 times, given how fast everything is. which brings us to the MKV model. So there's another one of the readings from this week, right?
I have a super quick question about
Yeah,
bootstrap.
we can get back to that question later. Yeah, go ahead.
So like in that tree you have drawn behind you with A, C, and B.
Yep.
If you had like a bootstrap support on like the node at the bottom, is that? Oh, so I was looking at the one up higher. But
yeah.
Regardless, does that say is that reflect how often A, C, and B are a clade?
Yes.
Or does
So
it say something about the relation between A, C, and B?
so there's bootstrapping and how we summarize bootstraps, right? So we don't. I mean, think about bootstraps as like being on nodes, right? That's how you always think about it. But what does it actually give us?
The whole tree.
A tree, exactly. So I get right, get this set of trees, right? And I want to summarize those, so I want to do a consensus approach, right? Or I have a best tree and I can figure out. Well, let's say I have my best tree. So there's two ways to present it. The way you normally do it nowadays is you have your optimal tree, right? Which let's just say is this. And I want to put bootstrap support values on this tree, right? So actually what there are, they're not support values for a node. They're support for an edge, by partition, basically. So it's saying, draw. So I can imagine this bipartition, right? Which just corresponds to, well, BCAD, right? So that means this edge separates BC and AD. If you think about it, bootstrapping is done on unrooted trees, basically. And so the unrooted tree, I have, this is the same as BC AD, right? And so it just tells me I have this bipartition. And so I get one count for that. Let's see here. So that's from this tree. Do I have that here? No? Do I have that here? Yes. Right, so I have two of those. Right? I can have other bipartitions. And this is actually a pretty simple tree because we add E to each one of these. Right? I still have that count. That counts right. What about AD, BCE? How many of those do I have? Everyone agree with one? Some of those aren't sure. Right? If you're not sure, raise your hand. All right. So, what do I mean by bipartition? Basically, is there a line I can cut on the tree that breaks into two parts? Right? And when I cut it, just list the tax at each part. Right? So here, I can cut this tree right there. Right? And now I have two components. I have one that has B and C, and one that has A, D, and E. That makes sense? Okay, so that's the bipartition. And so all I do for bootstrapping is we have our set of trees, and we're summarizing them by counting these bipartitions. Right? So we're saying we have, BC versus ADE. So is there a place I can cut such that one part that's all stuck together is B and C, and another part is all AD and E. And I don't care what the relationships are between AD and E. All I care about is that one branch I'm cutting. Okay? So ADBCE. So which of these trees has that? Bottom one, right? Because I can go slicing right there and get it. Okay. What about DE, ABC? First one, exactly. Right there. Okay, so I have one of those. CDE, AB. How many times do I have that? One. Good. So in the label my tree, what I can do is go on each edge and say, okay, for this edge, that corresponds to BC line ADE, right? Put that up, okay. That happens 2 out of 3 bootstrap replicates, so that's 2/3. Can't write anymore, okay. What about this edge? What do people think? Which bipartition does that correspond to, this edge?
The third one.
The third one, right? DE, ABC. Okay, I saw that only one of my trees that has a bootstrap of 1/3.
Where's the denominator coming from?
How many bootstrap replicates there are, right? How many trees I got out from my bootstrap search?
Oh, so the optimal tree doesn't count.
Right. Yeah. So optimal ES trees. Okay. So now I have. So now out of our plots, let's say 0.67, 0.3. What about this one? So a trick question, right? Because I don't actually have an output. There's no actual branch here. So it's actually just a trivial bipartition, right? It's just E versus everything else. So of course, right? Now these are rooted trees, then yes. If it's non-rooted trees, then there's no information about that. Right? Saying how do I have A as A all the time, right? So it's not bipartition. It is technically bipartition, but it's E versus A, B, C, D, which of course you're going to see all the time. Right? So that's what these numbers are. So these actually numbers, so like 0.67 corresponds to seeing this bipartition. Colloquially, we often say this node has a support of 0.67. But actually what it is this edge has a support of 0.67. Right? And if you want to summarize it, you could summarize it different ways, such that we look at like a tripartition and count that, and then it actually corresponds to nodes, but no one does it that way, so that'd be confusing.
So it's not necessarily support that that's a clade, because you can have that partition without it being a clade.
Right. Yep, because it's unrooted. Right. So, I could have unless you store these as rooted trees and have like an outgroup or something. Yeah. That's why it's not support for the clade, it's support for the bipartition. Right? Now, colloquially, if you force an outgroup and so it all has the same rooting, you can treat that as a clade too. Right? Yep, good. One other thing to worry about in this case is how we summarize it. Support can look worse than we think it is. So, let's say I have A, B, C, D, E, F, G, H. Right? And let's say I have one taxon I that can go either here half the time or here half the time. And everything else stays constant across my bootstrap trees. Right? What's my support then for this bipartition? All right, so half my bootstrap trees have I here, and half have them here. Right, so for this edge, half the time I's on this end of it, half the time I's on this end of it. Right, so bootstrap support for that will be 50%. Right, that makes sense? Even though all these relationships don't change. Right, so this is how we're summarizing bootstrap. Right, so we're summarizing it by looking at these bipartitions. So one taxon hopping around makes everything look bad. Right, if I summarize it a different way, right, it would look perfect. So there's ways of doing called maximum agreement subtrees. We say, what's the tree that's present in all these trees? Well, it's everything but I. So just cut I off. Right, well, 100% support for that. Right, and so some people talk about like rogue taxa, removing rogue taxa that improves support. You could just be deleting this one I taxon that's hopping around. It doesn't actually make the tree search any better. It's just that your summary looks like this. right? So it's a subtle thing that we often ignore, right? That can matter. And same thing, we summarize Bayes trees the same way, right? And we could have the same issue here happening. Okay, so it's worth distinguishing bootstrapping versus how we summarize bootstrapping. Right? And so you could summarize it as, quartets, right? So 4 taxon trees. I break this tree up into a bunch of 4 taxon trees, and like the 4 taxon tree C, D, E, F is always present. 4 taxon tree A, B, E, G is always present, but I, A, F, G is present only half the time. So you can summarize it with quartets instead. Right? But we don't do that in the field. That's another thing we can develop a new method of summarizing. And so one of my colleagues actually, part of his dissertation was taking these trees and figuring out ways to prune off taxa to get a subtree that showed higher support, not in order to skew the results, but so you could actually see, oh yeah, there's this core we actually know something about. It's being obscured by these things hopping around. Right? But it's actually a mathematically very hard problem to do. So, yeah. So
On a package now.
now, it was I think it was a C program, but no one uses it. So, yeah. But again, it's something you could just so what you could do is just implement that in R. Oh, cool! I can figure out the core goodness of my tree. Use it as a rogue taxon tosser or something. Yeah, because this matters. Because we care about getting good support for trees, and it could be that a few things are hiding the support because of how we summarize. Good. All right, so that's bootstrapping. Right. So, what's a good value? So, Jen, you said you want to get good values for bootstraps, right? What's a good value? 70, right? That's what everyone says, 70. Why do we say 70?
Because you're over 2/3.
Because Joe Felsenstein was married. No. Yes. No. There's actually a paper by Hillis and Bull, and they did simulations to look. I think it was simulations to look at what bootstrap value corresponded to having the bipartition be accurate 95% of the time. It was 70. Right? So that's why we get. So for their simulation study, that's what they got. So that's what we got. That's what we made our own rule of thumb. Right? Is that still the case? We don't know. No one's come back and looked at that. Right? So what you could do is do analysis where you say, okay, let's say I have next gen sequence data. What's a good value there? So if I simulate this as should be there 95% of the time, what bootstrap value does that correspond to? Right? Let's see. Right, that's where that rule of thumb comes from. And then it is true, that's what people usually do. Yep. Whereas Bayesian posterior probabilities are often higher than that. Right? But again, remember, they're measuring different things. Robustness versus probability given the data I have exactly. Other thoughts about this? Okay. What about that Lewis paper, the one on the MKV model? Little questions about that? People read that? I see one nod. Good job.
The Lewis 2001.
Which one? There's actually two. Yeah. Yeah. He had a really good year. Yeah. So the likelihood model one. So any paper like that's worth thinking about. What's the problem he's trying to solve? Why is he doing this? And so what? Right, what kind of discrete data? Morphological. Right, and why is morphological different than DNA? It's all two bases with 2 states, 4 states, whatever. What's the difference?
One difference is you don't have invariable sites.
Right. Right, so I'm trying to do a study of mammal phylogeny. Right, has hair. I'm going to go and check all my 1,000 specimens. Do you have hair? Yep. Do you have hair? Yep. Do you have hair? Yep. Well, I know they all have hair. Do you have bones? Yep, yep, yep. So when people actually code these data, they don't bother putting in the invariant sites. the thought is like for parsimony, invariant sites don't matter at all. Right? They don't affect the score. So why waste my time recording them all? Right? Likelihood models they do. Why?
It affects the rates of change that are happening along those branches.
Exactly.
If you include those invariant sites, then you can have that data that gives you the number of changes, even if they're unique to the clade.
Mhm.
They will influence your branch lengths. Because parsimony doesn't do anything with branch lengths.
Yep, exactly. So we're going to talk about that when you have Bayesian with lots of data that are invariant, how that increases the possibility probability. Same thing, the likelihood model at the heart of it is saying, oh wow, the rates are really slow here. Right? So it actually uses that information. Whereas parsimony just counts changes. Right? And so if you use only variable sites for likelihood, the model will think that it has very, very fast rates. Right? Why is that a problem? So model's wrong. All models are wrong. So don't sweat it.
If you have a problem with long branch attraction, if you have like super fast rates just across the tree.
Right. So it could be you could have issues that. So likelihood can fix long range attraction, which we'll talk about in a little while. If you have the right model, but the wrong model, you have no guarantees. That's one thing. Yep. Actually, there's a paper in Science, which is really good in Science, a few years ago, showing how likelihood is subject to long range attraction if you had heterogeneity in a certain way, which makes sense, but yeah. Good. Right. So the wrong model, you get the wrong result, the wrong tree back out. Right. And so what's the solution? Conditional. Right. So what does that mean? Yeah, you're right. You're right. Conditional stuff.
Somehow you build that bias into the model.
Right. Exactly. That's all this basically is. And it's something that Felsenstein wrote about in the 70s, right? But then Lewis said, well, we should be doing this for morphology, so he wrote a model. Those MKV models. Basically, you're just saying, we take into account that we have censored data, right? And we do this all the time in stats. So if you're looking at life expectancy, you have to take into account the fact that if for people who are 30, take into account the fact that people died before that with censored data, right? I've heard about ascertainment bias with the dolphin example. So you're in the core, right? Do dolphins save humans? Well, how do you know that? So we have shipwrecked sailors who say, yes, I fell in the water, the dolphin pushed me ashore, and I was saved. Right? And it could be the dolphins say, oh look, a mammal, he needs to breathe air, let me push him ashore. Or it could be dolphins say, look, fun flappy thing, I'll push it somewhere. And only those that end up on shore survive and say, dolphin, help me. Those who are pushed the other way don't say anything about dolphins because they're dead. That's that ascertainment bias. And so we have ways to take that into account, right? And so all the MKV model is just a way of dealing with that ascertainment bias of we throw out invariable sites. Okay.
I think it's kind of funny how he says this never happens with molecular data, but now people that you just use SNP data and next-gen data, it's the same thing, right?
Yep.
You're only using variable sites.
Actually, I wrote a grant with someone who was here about SNP data and ascertainment bias and the risk there when you get funded. So we don't know what happens then. But right, exactly the same thing happens with SNP data. And so, and you could use the MKV model for SNP data, right? Or anything like that. Allozyme data, if you want to go old school. Right? So that's worth considering. Now, one thing about the Lewis model as written is it deals with only invariant versus not invariant, right? There's other things where you have, let's say you have 0, 1, 0, 1, 2, 0 as my site. Right? It's not parsimony informative, right? It's not clear whether that be included or not included, right? It has information about branch lengths, right? The ones that have different changes may have longer branches, but it end up being included in your morphological matrix because it's not parsimony informative. And so you have to put that as ascertainment bias in the model, right? Things like that. And RAxML actually has 3 different ways of dealing with this sort of thing. It has a Lewis model, has an early one from Felsenstein, which apparently differs a little bit, and has one that Stamatakis wrote himself, too. And I'm not sure about the differences between them, right? And so you all have happily ran the morphology thing, right? Using ascertainment bias. Do any of you know which model is the right one to use? No, this is a risk with phylogenetic inference, right? Where you're happily using this thing. This could be a really dumb model, right? It could be not modeling the ascertainment bias you have in your data correctly. It should be used for different kind of ascertainment bias, right? Yale happily did the homework, didn't complain. There were no emails, nothing on Gitter about it, right? And this is the big danger about using stuff in phylogenetics, where using stuff because other people do it, you don't understand it yourself, right? And that's the point where you should hand draw your trees and count parsimony scored on it by hand. But it's worth digging in to figure out this thing that I'm doing, that I'm basing a third of my dissertation on, right? Do I understand what's actually going on there? Right? Do I believe it? And so that's where going back and reading literature to see, oh yeah, that makes sense.
So as long as you're not on that committee, though, it'll probably be fine.
I want your committee there.
Well, hypothetically, or something.
Yeah, as long as people on your committee don't care about whether you're getting it right, you're all set.
Nice.
Yeah. Which is, you have to say, pretty good odds, actually. Yeah, I mean, it's also one of the reasons I, in this core, I love PAUP, but we're not using PAUP in the course, right? Because it's closed source. So if you really cared about this, you could you could dig into RaxML's code and see where you choose which model to use, what's actually being done in the code there, right? And it'll take you a while at this point to figure out to grok what's going on, right? But you could, in theory, do it. You could say, hey, Fordyce, show me what does this mean here, right? You can try tweaking it. And what happens if I do this? So you can go in and dig it. Whereas PAUP right now, you can't go in and check that stuff. Right? It's all closed. So as a paper describing it, fine. If it's not, you don't know what the algorithm actually is internally. Right? And using methods uncritically can, oftentimes methods aren't testable. So at the end of the semester, we'll talk about method testing, right? But there are methods. There's a paper that was published in Nature, looking at a method for delimiting species. Right? So if I have one gene, I can split that. I can use that one gene. I can split these 60 things into 30 species. Right? Wow, there's actually many species. And nowhere in that paper they checked to see whether it worked or not. They didn't say, okay, let's imagine the method. Let's have this thing where we have known species and see if it picks that up correctly. Right? That wasn't tested. Let's simulate with a certain species model and see if it picks that up. Never done. Right? This method is still published and out there. Right? And other people use it too. I got a bunch of papers to review that were, are there species in asexual taxa? Well, yeah, because this method tells us there are species in asexual taxa. It's giving you an answer is the true answer. Right? It's very easy to fall into that, right? Where you use something uncritically in science, but you have to be careful. Right? Not that people are out to trick you or have lying results, but just stuff could be wrong. Right? Questions about that? Okay. So one question is, so now we have parsimony for dealing with invariant site, dealing with morphology, but likely for dealing with morphology, right? How do you choose which to do? Right? Or in your case, or if you're using in neontology, you could do parsimony, likelihood, Bayes, neighbor joining. There's a whole variety of methods you could use. Right? What's your thought process behind choosing something like that?
Which one gives you the answer I hope for?
So you don't want to be Bayesian then. It's wrong prior. Yeah. Prior probability age of the Earth is 6,000 years old. Let's see. Ah, yeah, found it. I thought of doing that. It'd be like going to the dark side. They didn't get 10 years, going to publish a paper. Yeah. Right. I mean, it comes back partly to why you're doing what you're doing. You're trying to get the best tree, right? You can try to figure out which methods are most likely to give you the true estimate, right? The best estimate of the phylogeny, right? Another school of thought that says what we really want to do is be philosophically pure, and so it's a very small school of thought, but there's a reason. It's worth taking a break to show you this. So let's hopefully summarize it. So There's a journal called Clitosics. And so there used to be in the 70s and 80s, maybe 90s, this sort of religious war between people who wanted to use models on one hand, people who wanted to use just parsimony on the other, right? And that ended up that the model people basically won, but there's this subset of people who really care about the philosophy. And so, you know, this journal, if you want to submit to it, they say, you know, there's strong philosophical arguments in terms of using parsimony for inference, right? And a tree on parsimony alone is sufficient in this journal. So you want to publish in this journal, you don't need to worry about any, you know, likelihood or Bayes. Just do parsimony, we're happy with it, right? And it's based on the philosophy, right, of science, right? So there is that fight out there, right? What would be a counter argument for this? Something you read.
Just because it makes philosophical sense doesn't mean it's like biologically motivated. It might not match up with what you think as well as like
Right. So you could say philosophically this is better, but in terms of like known examples, look, it gives me the better answer. Right. In terms of like, you know, humans are one species, not many species. So I don't care what parsimony says. What's pure, like, is maybe the best answer in that case. Something like that. Yep. Good. What's one example of a problem like that? Something you read. Oh, did they not have you read Felsenstein's zone? No, I tell you read this, yeah. So what's that paper about?
Sometimes two character changes is more likely than one.
Right. So the core of parsimony is what? What does parsimony mean?
Lowest number of changes.
Right. Fewest ad hoc changes. Right. I mean, generally means cheapest, right? So they have the fewest ad hoc hypotheses. Right. So the best parsimony tree is that which requires you to have to make the fewest changes, right? So, you know, here I have 1, 1, 0, 0, 0, 0. This requires, and this is 0. This requires a 0 to 1 change here and a 1 to 0 change here, say. Whereas a different tree that had these being clayed would require just 1 change there. So it'd be a better tree, right? So that makes a lot of sense, right? As a criterion, right? So like I have some trait, and I have eyes. So I have this tree says eyes evolved 15 times. This tree says eyes evolved once. I'll go with the one that evolved once. You know, wings. How many times did wings evolve? Probably once. Right? So what's wrong with that?
Those are rare traits, and if it's something that it's more switch up. Like if you're talking DNA, the bases are more likely to switch back and forth a few times in the direction than just once.
Right. So if you have enough changes, then you could imagine that you have 2 changes. That's actually better than having 1 change. Yep. And so this Lambert attraction issue comes up with in Felsenstein's paper on positively misleading. Right. So what does it mean positively misleading?
It's telling you that it's the correct answer, but it's actually taking you down the wrong connection because of that long range attraction. The multiplicity of changes that are happening in the separate branches are actually tying them together, bringing them into an assumed relationship that is not factual.
Right. What happens if you add more data?
Well, it's a further number of changes that actually reinforces that relationship that's false.
Exactly. So like if you're doing something like a Bayesian analysis, right? We'll talk about a little bit more next week. The idea is you have your stupid prior, right? As I add more data, eventually it outweighs the prior. Right? I get to the truth, and it drives me to the truth. Here's not Bayesian, but here's the opposite, the reverse problem, the reverse issue. So as I add more data, I get more and more confident in my answer, which is still the wrong answer. Right? There's lots more data for my wrong answer. Right? That's scary. We hope, okay, I can say like, yeah, of course, I have finite data, I get the wrong answer because I just don't have enough data, whatever. Right? That happens. But if I work really, really hard, I get the wrong answer even more confidently that's not something you can it's not wholly complete yourself out of with more data. Right? So why does that happen? So reading the paper, talking about long branches. Right? What does it mean about the long branches?
Homoplazy.
Right. So what's homoplazy?
Homoplazy is the origination multiple times of a similar or same feature.
Mm-hmm.
Sometimes you can correct your information by better characterizing what those characters are and see if they really are homologous.
Mm-hmm.
But otherwise, this problem still exists. And so homology, on the other hand, is those features that organisms share because they have a shared relationship.
Mm-hmm.
And that's evidence of that shared relationship.
Right. So like looking, for example, at ichthyosaur tail fins and dolphin tail fins, right? They're both flat, like, and they push the water, right? And they look pretty similar, right? And so you can score it as, dolphin, 1 for tail fin, ichthyosaur, 1 for tail fin, dog, 0 for tail fin, bird, 0 for tail fin, right? But the idea is if you look at it close, you can say, oh, wait, the dolphin tail fin is horizontal, the ichthyosaur thing is vertical, right? So actually they're not the same stage. So it'd be 1 for dolphin, 2 for ichthyosaur, 0 for dog, right? So that's the idea. If you look at it closely, there actually is no homoplazy, right? And that works maybe for morphology, right, with these complex traits underlying it. But if you look closely at this A, you can tell it's not the same as that A. Like you can't do that for molecular data, right? This tyrosine is the same as this tyrosine, right? You can't get at that. Right? So there's that homoplazy. And even in parsimony, it can be hard to dig into it. It has something like, you have traits like large versus small pore. Like, okay, well, it's bigger or smaller than 1 millimeter. What about 1.3 millimeters? And is this 1 millimeter different from that 1 millimeter? They're not homologous millimeter lengths. Maybe that falls apart too. Right? So even with morphology, it can fall apart. Right? So then, what would be an example of a tree that might have this issue? So I'm going to draw one. All right, well, long range attraction, right? That's the colloquial term for this. What does that mean? What are they attracting? Other long branches, right? Why?
Because each one has had a long time to independently evolve as a tree.
Mm-hmm. And so homoplazy might occur in those branches, right? So, and what does it mean for it to be long anyway?
Means that there are a lot of changes along that branch.
Mm-hmm. Right. So, for example, we could have this tree, right? And this could be flies, elephants, sequoias, ragweed, right? So short generation time, long generation time, long generation time, short generation time, right? So if we think that the rate of evolution might be proportional to the number of generations, right? These would have longer branches. Okay. So what? How does long construction happen on this tree?
You're going to say those are A, B, C, D. You're going to get A, D, B, C.
Mm-hmm. Right. So I was so long branch attraction would say, the tree I might get out might be right where A and D are bipartition. Right. So that's what the end result is. Right. How does that happen on this tree? Yeah, let's do it. Well, could do rooted or unrooted? Doesn't matter for this. Let's imagine it's unrooted. Right. Okay, let's evolve a trait up the tree. Okay, so here we have a trait. I start in state. Here we have a trait in state 0. Right? What would evolving the trait mean? I'm going to write a simulator in R for this. So it's a good exercise. What would you do? Why don't you break up and talk about this? So, read through the paper a little bit and talk to each other about how you would simulate this and what it would look like. Right? I don't think you're quite getting it. I don't want to just barrel through it. Right? Because this is a key point.
You're talking about the Gaussian
Yeah.
What did folks think about all this? What did you come up with? So first of all, how do you simulate traits on this tree? Maybe you just like write an R script or something. What does a long branch mean? So this is why sometimes we write simulations so you can actually show like, okay, like long branch, you think you know what you're talking about, but you're going to say, okay, write code for it. Like, oh, what does actually that mean? Right? This helps encapsulate what you know. So if we were to simulate long branches, well, how would you do that?
You would, for each of those nodes, you're going to, if we're going to make A and D have a long branch, you say that their rate of change is going to be higher than C and E. And you can also say that you know that A and B are related, and C and D are related, but actually that's outside of it. You just put in these rate changes that are different, and then you see how that affects things. You can put in that parameter.
So let's work through it. So let's say, okay, I start at 0 here. I'm going to get something here. How do I tell the computer to make something here? So it'll either be 0 or 1 here, right? What do I tell it to do?
Rate times time.
Rate times time. Okay. So I have, let's say my rate here is 1, and my time is 1. So rate times time equals 1. So now I tell the computer 1. Yeah. What next? 1 what?
One opportunity for change.
Okay. So how would you do that?
Then you can give it a probability of the change happening.
Okay, probably change happening. All right, so go back to our coin flipping thing, right? So you could say, I start with 0. What's the probability of having a change? And you can say that probability of change prob change equals what? I don't want to say 1 here, right? Because then we must change. Maybe that rate's a bad rate.
Exclamation point, prob, not change.
So you want them to be equal? Well, I mean, it's true that the probability of change is not the probability of not changing. That's good. What are you actually doing? You're trying to say, I start with 0, end up with 0 or 1, right? Just like coin flipping, right? So just because he's like, I start with heads, is it going to end up in heads or end up in tails? Right? I have to do what? All right, so if I said, write an R script, I give you starting state is a function. So foo is my function. I have starting state, time, rate, right? give me return state. Right? In here, write something.
So you simulated a coin flip
Simulate coin flip.
given your grapes.
So how did you simulate coin flip?
Draw
P.
0,
P.
1.
Draw 0, 1, right? What probability? Is it going to be a fair coin or not?
Depends.
You're saying no. Why not? No, Christian, you're saying no. Why?
0, 1.
Right. I'm going to say 0 means no change, 1 means change. So flip a coin. If it lands heads, I change the state. If it lands tails, I don't change. Right? On a longer branch, there's a chance of being heads B. Relative to a shorter branch.
Between the node and the tip of the branch, not in for a given amount of time. In the total branch,
Total branch, yeah.
probably is higher.
Probably is higher, right? So the longer the branch, the greater chance of flipping and getting a change, right? So here, let me do some R code quickly.
But how would you be able to predict that before putting it in the code? Which one needs more weight?
Well, let's work through that. So, can you see this? And actually, you can make it simpler. Just have probability of change in the branch. Right? Yeah. Just some bookkeeping. Right, does that make sense? So we say, you have our starting state, you have probability of change. If something happens, right, then we change our state. Right? Otherwise, final state is the starting state. Right? And just to keep it, rather than having to do complex tracking of what the starting state is and stuff, I can just do, you know, if it started off 1 and goes up and changes and goes up to 2, so just make that back, so I'm just going back to 0, going binary traits. There's other ways of doing this too. But the main heart of this is this, so if do something. So if I have a change, I'm going to change my state. Right? So how do I decide whether I have a change or not? How do I decide if I have a change on this branch? So what's happening underneath this? And this is just like Felsenstein's paper, actually. Yeah. Nope. Not the likelihood in terms of like the likelihood equation. In terms of just some parameter, though.
You just do a random draw, and if you get like 0.01 or less, then you change.
Yep, exactly. So why 0.01 or less?
Because that's what you, other trail shows up there.
Right. So if, you know, RUnif 1, 0, 1, so draw one number between 0 and 1, if that's less than prob change on this branch, then change. Right? So think about it. If my probability change was 1, right, this is always going to return yes as a change. Probability change is 0, what happened? Always returns no change, right? Because something is returning a number. If probability change is 0.5, half the time it picks a number above 0.5, half the time it picks below 0.5. Right, so this is exactly what you want. Right? Does that make sense? No? Okay. Ask questions. Yeah. Let's go here. Here I have a uniform interval from 0 to 1, right? So our unif just picks a random number between 0 and 1, right? So let's say, pick a number. What? Yeah. Okay, I was thinking the same one. Weird. You pass the house. Right, so pick one at 0.3, and pick another one here, another one here. Right? What fraction of them will be 0.3 or less? What do you think? No, I don't have a good sample.
3/10.
You say 3/10. Why?
Because you're asking for the probability being less than 3.
Yeah. And it might be easier to think about it like just integers from 1 to 10.
Because 3/10 of the options are
Right, so I just pick numbers at random. I'll expect to pick this one 10% of the time. Actually, 11% of the time. Oh, no, not 1 to 9. Oh.
We're starting at 1, not 0.
Yeah. So 10% of the time we get a 1, right? 10% of the time we get a 2. 10% of the time we get a 3, and so forth. Right? It's picking evenly, right? So what's the chance of getting something below 3.5? 30%. Below slightly more than 5. Right?
So 20% or below 3.
Right. And so that's the discrete numbers. Continuous, let's do lower than or equal to. Right? So I'm not going to get exactly 0.3, but I can get really, really close. Right? It's continuous. The same thing. Right? Going here, 30% of the numbers are below that, 30% are above that. Right? Here. I'm doing it. So just this, doing 100 numbers between 0 and 1, right? And I could plot a histogram of this. And actually it likes the big numbers. Let's try to give it a larger number, right? To get this sort of flat histogram, right? If I were to do the quantiles,
Random uniform.
So R whatever is generate a random number from some distribution. So I could do R norm to generate a number from random normal. I could do R unif to generate a function from a uniform distribution. R binomial to get something from a binomial distribution. Right? And so the uniform distribution, the secret thing underlying all stats nowadays is Wikipedia. So yeah, it looks like run it.
It's an argument.
Yeah. Yeah, so here's a uniform distribution. So like within this range from A to B, everything has an equal chance and nothing outside that range. That makes sense? Okay. So, and you can see, so I just generate a bunch of, so that's what generating randomness between 0 and 1. And so how many are at 0? Very, very few. right? Below 0.1, so that's 10%. 20% is about 20%. Right, so this is flat. Whereas if it were a normal distribution, it's sort of symmetric around 0, right? But you see this is much further out than this is from this. Right? I could plot that. Something like that. So uniform is, even sampling numbers evenly. Okay, so if I'm sampling numbers evenly from 0 to 1, then half of them is going to be below 0.5, half of them is going to be above 0.5, 30% are going to be below 0.3, 30% are going to be above 0.3, and so forth. Is that good? Okay, look good. Okay. All right. So here's what we're doing. We're saying, the probability of this change in this branch is really, really high. 0.99. The 99s out of 100, I generate a random number in this interval, it'll be below that, right? And so, it was below that, it's good, it counts as a change, right? And so, what does that correspond to in Felson's paper? Here's a variable for this. So, we call this probability change on this branch. Here's a more succinct variable. Yep, exactly. Or Q, depending on the branch, right? So in the short branch, he calls it Q, and the long branch, he calls it P. There's a probability of change. That's it. Okay? And so that's what we implemented here, right? So we can try this. And two 0.5. Find a good mixture of zeros and ones, right? Oh, it's not good. Okay. Enjoy your seed issue. We get a good mixture. Whereas if we have a really low probability of change, we almost always get 0. Right? And so that's what you would do. You would simulate, you'd say, I start here at 0, right? And to get my probability of change, sometimes I get 0, sometimes I get 1 here. Okay? And then do the same thing. So let's say I get a 1, let's say I get a 0 here. And now I have a bigger probability of change, right? He calls P, right? And then I might get a 1 here. Okay? And here it could have, and on the E it could happen to have no change. This way it could have no change. This way no change. Maybe this way also no change. So what does this character tell us about the phylogeny? Under parsimony. What?
That character only tells us that there's been a change on A.
Right, so there's been a change in this branch. It doesn't tell us where A relates to D or E or B or C, right? It has no information for parsimony. Okay, so no matter how many times that gets generated, won't affect the trees we get at. Won't cause long range traction, right? What if it were 0 here? What happened? Does that affect the trees? Score of the trees? No, right? Everything's the same. So no matter what tree topology I try, they'll have the same score. What if I had this? So my data matrix becomes A, E, D, C. Does that suggest a tree? So there's many possible trees. Right? Let's say AE, DC, AD, CE. Okay. So who thinks this is just tree 1? Raise your hand. Tree 2. Okay. Those things is 1. Explain why.
Well, in parsimony, it makes sense it was involved once in the branch before the node of an AE.
Right, so you think about the unrooted trees, it's often easier to do that. If I have AE, DC, and this is 1, 1, 0, 0, it suggests I have one change here, right? So a 0 to 1 change or a 1 to 0 change, correct? Whereas if I said, okay, maybe it's actually this tree, I have 1, 1, 0, 0. How many changes does this require?
At least 2.
At least 2, right? And they could be, here and here, right? Or it could be here and here. I don't know which it is, but either way, it's 2 changes, right? So parsimony prefers this tree 1 change better than two changes, right? So that's cool. That then gives us this character suggests that we should have the true tree. Great. Want to see that? Okay. And of course, it doesn't matter if this is a 0 or a 1 here. If there's a 1 here, then we have 1 change in truth. If there's a 0 here, we actually have homoplasy, so homoplasy breaking our way. Right? It's still giving us the right answer. Right? So, awesome. Right? Okay. What if it is this?
You get that tree on the right.
If we have this tree on the right. Right? Because now we have A, D. So this tree has one fewer change than this tree does, right? Change versus change. So then parsimony says we should have this tree rather than the generating tree. Right? And this goes back to Felsenstein's argument. Right, so here he draws the trees a little differently, right? So he does it as a rooted tree, but still with 4 taxa, right? He's doing this way because it's easier to see. And so he has all this math, right? And I guess he goes through, and what we're getting is the probabilities of these patterns, right? So in his case, the root's always 0, right? And okay, so he has these probabilities for 0, 0, 0, 0, 0, 0, 0, 1, 0. So the 0, since he assumes the root's 0, he doesn't show it, right? But basically, you get these probabilities based on P's and Q's, and R is that length of the root of that other edge. Okay, and what you can do with that is figure out what portion of the time I get this or this, right? Because this one gives me the truth. This one gives me the other tree. So if we generate more of this kind, awesome, we get the right answer, right? If the branch length is set up such that we get more of this kind, we get the wrong answer for parsimony. Right? Does that make sense?
So what's informing the branch lengths, though?
The truth. So out in the world, there is a certain rate of evolution, or a certain amount of time. Right? They're saying if your true tree has these branch lengths, all you see when you're doing the analysis is the raw character data. You don't know what the truth is. So what you do is from the character data, you try a bunch of trees. Right? So if we have more of this kind of character than this kind of character, I will then choose the true tree. Right? If I have more of this kind, I'll choose the other tree.
in this case, all of the characters are evolving under the same reins, P, T, and Q.
Right. And of course, we also have characters like that one. I don't care. Right. I don't care. I don't care.
But so if you added more characters, though, wouldn't that decrease the probability that you would end up with the wrong tree?
So that's a good question. So if I have, if the tree, generates 90% more of these than this, right? So many more of this than that one. But I have a small sample that only have one character. Then I could by chance get this, right? Part that makes this scary is if it's more likely to get this misleading character than this one, I might luck out and get the true the useful character first. But as I add more characters, the chance of having more of these and these, grows to 1, right? And so then I'll be guaranteed to get mostly these characters, and then I'll get the wrong tree. No? Yes? No. Okay. Maybe see my book.
Point that with the left tree, if that is the true tree, we are more likely to get
This.
data that looks like the wrong.
Exactly. So the so-called Felsenstein zone is this area. So it's actually So if your branch lengths are in this area, so here's P, here's Q. If your branch lengths are in this area, you get more of this than this. So as you add more data, you get more and more characters that support the true answer. You're good. Right? Awesome. If you're in this area, right, this this character pattern is much more common than this character pattern. Right? So as you add more data, you get more of this, and so it looks to you like it should be A, D, E, C. Right? And that's the Felsenstein zone. That's where more data don't rescue. So in this case, right, we can say, all right, what's the probability of having 2 changes here versus having 1 change here or 1 change here? Right? So if having a change here, let's just make this a loopy one so it's easier. But it's nice for an unrooted tree. I have to change somewhere on this edge, right? So I have one probability of a change on this edge, and then probably a change on this and this. Right? So I have 1 change on the tree, parsimony works. If I have 2 changes on the tree, parsimony gives me the wrong answer. Right? So the question is, when are these branches long enough such that 2 changes here are more likely than 1 change here? Does that make sense?
They've got enough time to have that change happen.
Right.
It depends on the ratio between P and Q.
Mhm.
Difference.
Yep. Yeah, and also the absolute value too. Because it's just the ratio. This would be a linear straight line. It's curved. So. Yeah. Does that make sense now? So, if this branch is short, or the branch that gives you the true homology is so short you have very few characters evolving there. We have a lot of characters evolving with homoplazy here, right? The input from this is much more than from this. And so parsimony will say, oh, I see these changes happening more, so I can minimize my total score by moving A and D together. Right? And that's the Felsenstein zone.
So I have a weird question.
Yeah.
What would this tree look like if we do it ultrametrically?
Oh, so if you made it like if you did like a clock model or something, it would make it such that A and E were the same height as C as D, right? But you'd have to you'd hear the branches screaming as you pulled them. Like, no, I don't want to be longer, right? But you can force it. Yeah. Right. And so parsimony in this case, if you have these long branches, can be misleading. And of course, sure, you can imagine this that's crazy talk. Yeah, in theory, use a weird corner case where if this branch is a billion times longer than this one, yeah, I can see that, right? But in real life, does it matter? That's a legitimate question, right? So, an early paper, the only paper that actually coined the term Felsenstein was by Helsenbeck, and there's a case of Strepsiptera. Right? Who knows what Strepsiptera are? You should. Okay, what are they? Yep, just living parasites. And these really cool things where the females are these like little things that just grow out of like a bee abdomen sort of thing, and have no eyes or anything. And the males have these really weird. I have the web.
You're like your just sort of dance.
Yeah. Yeah. So they have these really cool. So here are the females, like just sort of hidden in there, and the males have these really cool eyes, and they have these things that look. So you know how flies have halt ears, right? Behind their wings, they have halt ears. So they have halt ears in front of their wings. Okay, and so the question is, are these halteria things? Are they like reduced elytra of beetles, sexually related beetles, or are they homologous with halt ears? Yeah, sorry. And just it just reversed, right? So it has some like Hox gene mutation or something, right? And flies have, if you look at rates of molecular evolution, flies are much longer than beetles. So anytime you look at an insect tree, you'll see, there's the Diptera, right? And Strepsiptera were also apparently a long branch. And so they're coming out with flies, but maybe they should have been with beetles. And the question was, I think the paper was held like, is a Felsenstein zone a fly trap? Right? So are these Strepsiptera being like sort of drawn in to flies where they both have long branches rather than due to actual relationships? Right, and that's where parametric bootstrapping comes in handy. So you can say, if I think the relationship is truly Coleoptera, Strepsiptera, Diptera, right? And if I simulate under that and I get back out Strepsiptera, Diptera, Coleoptera, right? So if the truth is this, and I simulate under this and I get this back out, it suggests that it's a region where, this long range direction thing is real enough to be a problem, right? In this case, that's what happened. Right? So if you simulate under these conditions, take the best fitting model that has what we think is a true tree, with all the branch lengths and stuff, and then simulate, you get this model back, this result back out approximately. Right? Suggest then if this is the truth, you get this. Of course, if this is the truth, you get this too, right? So you'd hope, right? But it suggests that regardless of what your data were, you would still get this answer. It could be a long range attraction issue, right? And so this actually could be a problem in real life. Okay, is the concern. So why is likelihood escape this? This is used as an argument for using a model-based approach, right? So, so we can see our parsimony have a problem. We see why distance would have a problem. Why would likelihood escape this? What information is likelihood used that parsimony doesn't? The rates, right? And it has information from the states that don't change, from the characters that don't change, or the characters that have changed only once. It gives us information about the rates. Right? So with likelihood, if I actually doing this matrix, I'd probably see a lot of stuff like right? All this information that suggests that, wow, it's really badly drawn, along D and A, just as long as some long branches right there, because they have these autapomorphies, right? So it suggests that the A branch is long enough that you have things that have changed there and nowhere else. Right? So when you get that is by having A be a fairly long branch. And so likelihood then knows, effectively, that the A branch is long, and the D branch is long, and the E branch is short, and the C branch is short. right? There's information about those branch lengths. And so given they have other information, it's not quite doing it this sequentially, but it's a way of understanding it. So given that A branch is long and D is long, just having two changes there is pretty high, right? And so I could explain this trait by having two changes, which isn't that bad because I think it's already long branches with already lots of changes, right? And so that way you can totally take that into account and then not assume that these are homologies. Okay? And that's why I'd like you to escape this if your model is right. And that's an important caveat. If your likelihood model is not a good enough model, you can also have long retraction there too. You can undercount the changes. Okay. So ask questions. Not everyone on board, I think, so, which is fine. That's why it's a class. Right, so ask questions. What doesn't make sense?
So when you say you have to have the right model for that, you have to.
And it doesn't have to be that right. I don't think it's actually been shown how right it has to be. So we have it kind of like, if the model is the true model, you get it right. If the model is really bad in certain ways, you get it wrong. And of course, there's some line between.
What would be a bad model in this case, I guess?
Oh, so this is a science paper I was mentioning, where they have some data involving a model where A and D have long branches, and then under E and C have the long branches, and the swing model was only one rate for all the taxa. So that's a model, but it doesn't fit it. Yeah, so that's sort of heterogeneity.
So how do you do this with only variable markers or characters? So how does that work?
That's true. So
How do you prevent long branch?
I don't know if you can prevent long branch with the MKV model. That's a very good question. I mean, if you have odd apomorphies,
Right.
it would work.
So I guess
If you have
with non-parsimony.
Yeah, if you have parsimony informative, then I think you're still stuck. That's a very good point. That'd be a good little project to do. I mean, because people are using the MKV model in paleontology now. There was a Wright and Hillis paper that came out that showed that this works better than the MKV works better than regular parsimony for tree inference and morphology. But I'm not sure whether they used, just variable sites or parsimony informative sites. That'd be a really cool to look at. Other questions about this? And again, just make sure, why do we care?
This is obscuring our way into truth.
Right, in a way that makes you get the wrong answer. So you obscure in the way because you just don't have enough information. Fine, that happens all the time. If your method's telling you the wrong answer very confidently, that's a problem. And so you might want to avoid methods that do that. Right? That's the argument.
So then with this, how do you know if you're in a situation that you need to avoid parsimony?
Well, I mean, it's like, I only wear my seatbelt if I know I'm going to be in a car crash. I'm not going to be in a car crash, I'm not going to bother buckling up. Or I can say, I'll just wear my seatbelt all the time.
So to you, wearing the seatbelt is to never use parsimony.
Or to never rely on parsimony. I might, actually I won't. But yeah, I won't use a parsimony tree. But people do. It's like oftentimes you'll see papers that will have a parsimony tree and a Bayesian tree and a likelihood tree, and that's fine, right? And they might pick up something. And the question is when they disagree, what happens? So if you're, publishing ballistics, you have to use parsimony only. And if you think that, let's see, where is it? Oh yeah. If alternate methods give different results and the author prefers unparsimonious topology, he or she is welcome to present that result, but defend it on philosophical grounds. Right? So for me, I don't want to defend my result on biological grounds, right? And say given what I know of the data and stuff, I think this is the biologically most credible thing, not which what dead dude thought was a good model, right? But yes, that's why I would just use likelihood and Bayesian approaches. Yeah, because I don't know of a case where I don't know of a case where you think that parsimony give you a better answer than likelihood, right? You can see where they give you the same answer, or likelihood to be better, right, and avoid this problem. But it's like, again, like wearing your seatbelt. Like, okay, most of the time it doesn't matter at all. You can just do one, right? When it does matter, you like to have your seatbelt on. Maybe you can imagine a scenario where like my car gently rolls into the lake and the door is locked, and maybe for not having a seatbelt in that case, right? But if it's low enough probability, unless I'm like driving next to the lake, just wear your seatbelt. So I think it's that sort of question. Yeah.
That's the takeaway for the day: is wear your seatbelt.
Yeah. I have one of those seatbelt cutters. Yeah, I mean, there are reasons to do it. I mean, it's like in paleo now, I mean, since parsimony is still used a lot, I mean, some people want to see parsimony trees, so as a service to readers, you can say, hey, you might not believe in this model stuff. Here's a parsimony tree. And maybe they'll agree, too. They can say, you know what, you might fight over which is better. Don't matter none. Right? And that also makes your paper stronger. So you can do it that way strategically.
I know from looking at those some leaves of parsimony tree, they're going to do Bayesian analysis. That's their initial model.
So you have a starting tree. Yeah, to make your search good. It's going to help huge tree spaces. Be able to get a fast starting tree that's in a goodish neighborhood is a big advantage. Yeah, so we'll do that. And actually, one fun thing, I think it's simple in steel. It's toughly in steel. The no common mechanism model. So if you create a likelihood model that says on every branch, for every character, it has its own rate of evolution, right? It's a super parameter-rich model, right? I mean, like, think about it. Like, for these, for this 4 taxon thing, I would have, 1, 2, 3, 4, 5, 6 branches. Basically, it's unrooted, so let's say 5 branches. 5 branches times 1, 2, 3, 4, 5, 6, 7, 8 rates, right? So 4 different rate parameters, right? That's a hugely parameter-rich model. The way you can fit that. But the cool thing about that model is the results it gives you are exactly the same as parsimony. And even in terms of like having multiple equally good trees, it gives you multiple equally good trees. It's exactly the same as parsimony. And so basically, you can implement parsimony in likelihood framework using a model like that. Right, so if you want to do model selection, do AIC or whatever, you can throw parsimony in there and see how well a 40 parameter model works against a 6 parameter model. Right? Probably not going to win. Right? But it's sort of cool. You can put them in the same framework. Right? That's a fun paper. It's called the No Common Mechanism, or NCM model. Yeah. Other questions or thoughts about this? One thing I haven't talked about as method is distance-based methods, like neighbor joining or UPGMA and stuff like that. Those are still used in the bacterial world a little bit, but people developing trees and metazoa and stuff don't use them at all. They have the lumbar distraction problem like parsimony does, and they're just based on like grouping on similarity, so even the clades don't like them. So people don't use them. They might be helpful for getting a starting tree because they're fast, right? Or getting gene trees because they're fast, but they're really not ideal to use, so I'd advise not using them.
Is it what it sounds like? You just attach something to its the taxon with the next similar.
Yeah, and then work your way down. And then,
distance or something.
Yep. Yeah, that's one of the algorithms. And there's a bunch of different algorithms for it. And there's different ways of calculating the distance. You can calculate distance on a different, GTR model and stuff like that. But yeah, and just group them together. I mean, UPGMA is nice because it gives you a tree that's ultrametric, so same root to tip length for everything. So it'd be good. That sort of looks like a clock-like tree, right? So it'd be useful for starting that sort of thing. But yeah, they're fast and dirty. And now, I mean, RaxML is insanely fast, so you don't have to, back in the day for me, doing parsimony search would take weeks, right? And now, for like, a few taxa, 40 different taxa. Now, you can do, huge runs, 45,000 species on your laptop, which is crazy. So, yeah. Other questions about any of this? So again, back to the papers. There was also the other Lewis paper, it was a good year for him, 2001, about phylogenesis mass turns over a new leaf. And I was comparing Bayes and likelihood. I also have a video up about Bayes and likelihood. So we'll talk more about Bayesian for phylogenetic inference later. But any questions about Bayesian approaches in general right now? So we know what priors and posteriors are. That's good. Okay, if not, you can watch the video. Let's wait a little bit. Okay, and that's another thing. So there's been various fights about how to make trees. There used to be a fight between, people doing phonetics, so just lumping taxa by group by distance versus parsimony, and that fight was won by the parsimony people naming clades, right? There's a fight between likelihood and parsimony, and it's been mostly won by those who use models, but with some significant, remaining pockets of resistance. And then I'm waiting for a fight between likelihood and Bayesian. No one seems to want to fight, right? And people will infer their tree with likelihood method and then use a Bayesian comparative method on it, or vice versa, right? And I think what it is people just sort of being practical, right? So certain things you want to use priors, because I really think the age of a clade, I have prior information about that, I want to use it. Other things I don't want to use priors, like the the branching time prior, I don't know, right? And so, okay. And also I think people are just pragmatic, oh, this program gives me the results I want, and so I'll run this program. So Surface will give me this modeling of OU parameters, and so I'll use that. And if something is, and that happens to use a Bayesian approach, but if something else use likelihood, they just use that too. We'll see next week, and also in, I think, 2 weeks, use of priors in more detail. All right, anything else? Okay, sorry GitHub was down last night, but do commit and push and pull your homework so I can see it. And we're not using GitHub Classroom anymore, we're just directly forking with the PhyloMeth thing. GitHub Classroom was not working for us, so just do the forking from now on. And I'll give you, I'll reject your pull request and say good job or something if you do it well. And one thing to note is if you don't do a good job, I'll tell you that too, and I won't close your pull request, I'll leave it open so you can go back and fix it.
So do you have to do a pull request once you fix it?
Nope, just add a new comment like, hey, I fixed it this time. Yeah.
So when you push it, the pull request is.
I think the pull request will be updated, yeah.
Okay.
If you don't see that, then let me know, but yeah, should do it.
So once we've done an initial pull request, we have to fix it. Then anytime we push something, it's going to notify you.
I think so. We'll try it. And if you don't see me reply, then ping me and I'll look. Oh, and do use the getter. So if you have problems, don't wait till the last minute. Just say, hey, it's a problem. We can all help. Actually, speaking of that, the Windows people want to meet, and I can help trying to debug the RaxML install that we can. The rest of you can go to the next seminar. Right?
So
Good.
into that binary.